If you really want to understand why some people continue resisting cancer well into their 80s while others struggle much earlier in life, you need to stop looking only at chemotherapy or genetics. There is something else that researchers have quietly been studying for decades, something far more ordinary in appearance, yet deeply fascinating in biological behavior. It is a tiny black seed that many people have seen countless times in grocery stores without ever realizing its potential significance. Inside this seed lies a compound that, in laboratory research settings, has been observed to significantly influence cellular regeneration processes, including stem cell activity. These are the same cells responsible for rebuilding blood, supporting the immune system, and repairing organs. In some controlled studies, this compound has been associated with increases in stem cell markers suggesting enhanced regenerative activity. At the same time, other research has explored how it may interfere with processes that support tumor growth, including blood supply formation, without showing strong toxicity towards surrounding healthy cells in experimental models. Despite all of this, it remains widely overlooked in mainstream medical discussions. It still sits quietly on store shelves, passed over daily, unnoticed by most people. And once you understand what researchers have been investigating, it becomes difficult to look at it the same way again. A fair question naturally arises. If there is so much scientific interest, why is this not commonly discussed by doctors? The answer is not simple, but it generally comes down to two major factors. First is the issue of patentability. Natural compounds cannot be owned in the same way as synthetic drugs, which reduces commercial incentive for large-scale pharmaceutical investment in expensive clinical trials. Without those trials, medical guidelines rarely include them, and without guidelines, most doctors are not trained to recommend them. The second reason is historical perception. This seed has been used in traditional systems of medicine for thousands of years across different cultures. Because of this long history, it was often categorized as folklore rather than modern pharmacology. Only in recent decades have scientific studies begun to re-examine it with modern tools and controlled experiments. That shift in perspective is where our discussion begins. I'm Dr. Liana, an emergency medicine physician and researcher in cancer biology. Over the years, I've seen countless patients facing serious illnesses, and I've also seen something interesting. Some individuals seem to maintain better resilience than expected, not always because of medical treatments alone, but sometimes due to patterns in lifestyle and natural compounds they consistently use. Early in my career, I focused strictly on conventional interventions, drugs, procedures, and protocols. That is how medical training conditions you to think. But over time, I started noticing recurring patterns in patients who appear to respond better than expected. When I traced those patterns, I kept finding similar dietary and lifestyle factors that were not emphasized in formal medical education. Among those patterns, one recurring element stood out more than others, a particular natural compound found in a very old, very common seed. That is what this discussion is about. Because today, I want to walk you through something you likely were never taught in your routine health checkups. I want to explain what this seed is, what researchers believe about its active compound, how stem cell regeneration is understood in biological science, and why changes in cellular repair processes matter so much over time. I will also walk through research findings from multiple institutions showing how this compound has been studied in relation to inflammation, immune response, and cellular behavior. Along with that, I will explain practical ways people incorporate it into daily routines in traditional and modern contexts. But before going further, there is something important to understand. None of this replaces medical treatment. It is a field of ongoing research, not a miracle solution. And with that clarity, we move deeper into the science behind it. The seed in question is Nigella sativa, commonly known as black seed or black cumin. It is small, dark, and unassuming, yet it has been used for centuries in traditional medicine systems across the Middle East, Asia, and parts of Europe. Historical records show it was referenced in ancient Egyptian practices, mentioned in early medical texts, and used in various cultural healing traditions for general wellness support. But the reason modern researchers have become interested is not tradition, it is chemistry. Inside Nigella sativa is a bioactive compound called thymoquinone, which has been the subject of extensive laboratory research. Across published studies, it has been examined for its antioxidant behavior, its interaction with immune pathways, and its influence on cellular stress responses. Scientists studying this compound often describe it as biologically active in multiple pathways, meaning it does not affect just one mechanism in the body, but interacts with several systems at once. One of the most studied areas is its relationship with stem cells. Stem cells are not specialized cells. Instead, they function as the body's repair system. They replace damaged cells in the blood, gut, brain, and other organs. Without them, the body would gradually lose its ability to recover from daily damage. As people age, stem cell activity naturally declines. This decline is gradual but consistent, and over decades, it contributes to slower healing and reduced regeneration capacity. Some laboratory studies have observed that thymoquinone exposure may influence markers associated with stem cell proliferation and differentiation. These findings are still being explored, but they suggest a potential role in supporting cellular repair processes under controlled conditions. At the same time, researchers are also examining how this compound interacts with oxidative stress pathways, inflammation signaling, and programmed cell death mechanisms in abnormal cells. This dual behavior, supporting normal cellular function while disrupting harmful cellular activity in experimental models, is what makes it a continuing subject of scientific interest. And this is where the deeper explanation begins in the next part. To really understand why thymoquinone has attracted so much scientific attention, you first need to understand what modern biology has learned about how the body maintains itself over time. Every second, your body is breaking down and rebuilding cells. This process is not occasional, it is constant. Your skin, your gut lining, your blood, even parts of your immune system are in a continuous cycle of renewal. At the center of this renewal system are stem cells. They are not specialized for one function. Instead, they act like reserve units, waiting for signals that tell them to repair damage or replace aging cells. When your body is healthy and young, this system works efficiently, but as aging progresses, that balance slowly shifts. Stem cell activity does not suddenly fail. It gradually weakens. Fewer new cells are produced, and existing ones lose some of their responsiveness. Over time, this affects everything from recovery speed to immune efficiency. This is one of the reasons why wounds heal more slowly with age and why resilience to disease can decline. In laboratory research exploring Nigella sativa and its active compound thymoquinone, scientists have examined how this molecule interacts with these regenerative systems. In controlled cell studies, researchers observed changes in stem cell markers, particularly those associated with hematopoietic stem cells, which are responsible for producing blood and immune cells. One frequently referenced study used advanced cell tracking techniques to measure how stem cell populations responded when exposed to thymoquinone in a controlled environment. What they found was not a simple increase or decrease, but a shift in activity patterns. Cells expressing regenerative markers showed increased proliferation compared to untreated samples, suggesting enhanced regenerative signaling under laboratory conditions. It is important to be precise here. These are experimental findings, not clinical guarantees. They do not mean a supplement can rebuild organs or reverse disease. What they do show is that certain natural compounds can influence biological pathways that are already responsible for repair and maintenance in the body. Another area researchers have focused on is oxidative stress. Oxidative stress occurs when unstable molecules, often called free radicals, accumulate faster than the body can neutralize them. Over time, this imbalance can damage cells and accelerate aging processes. Stem cells are especially sensitive to this type of stress because they need a stable environment to divide and function properly. Thymoquinone has been studied for its antioxidant behavior, meaning it appears to influence how the body manages oxidative stress in experimental settings. Scientists believe it interacts with internal defense systems that regulate molecules like glutathione and other protective enzymes. These systems are part of the body's natural cleanup and repair process. What makes this interesting is not that it replaces these systems, but that it may support them under certain conditions. In biological terms, this is called modulation rather than substitution. It is not adding something foreign, it is influencing existing pathways. Now, alongside stem cell and antioxidant research, another major area of interest is inflammation. Chronic inflammation is not the same as short-term inflammation, which is part of normal healing. Chronic inflammation is a long-term, low-level activation of the immune system that can quietly affect tissues over time. In many biological studies, thymoquinone has been observed interacting with inflammatory signaling pathways, including molecular systems involved in immune response regulation. Researchers often examine markers like NF-κB, which plays a central role in inflammation signaling. In laboratory models, thymoquinone has shown the ability to influence the activity of this pathway, which is why it continues to be studied in relation to immune balance. What is important to understand is that inflammation, regeneration, and immune response are deeply connected. Stem cells do not function in isolation. They operate within an environment shaped by inflammation levels, oxidative stress, and cellular signaling. This is why researchers do not look at thymoquinone as a single function compound. Instead, they study it as something that interacts with multiple biological systems at once. And as these studies continued, another question naturally emerged in scientific literature. What happens when these same pathways are observed in disease environments, particularly in cancer research models? That is where the next layer of findings becomes even more interesting, and it leads directly into how researchers began studying its effects on abnormal cell behavior, blood vessel formation, and programmed cell death mechanisms. We will continue from here in the next part. As research expanded beyond basic cellular repair, scientists began asking a more difficult question. How does this compound behave in environments where cells are not functioning normally? That question led thymoquinone into cancer biology research, where its effects were studied in controlled laboratory and animal models. To be clear, this does not mean it is a cure or replacement for medical treatment. What it means is that researchers were interested in how it interacts with specific biological processes that are also involved in tumor development and progression. One of the first areas examined was angiogenesis. Angiogenesis is the process by which new blood vessels form in the body. Under normal conditions, this is essential for healing wounds and maintaining healthy tissue. However, in cancer biology, this same process can be exploited by tumors to support their growth. Tumors cannot grow beyond a very small size without a blood supply. They require oxygen and nutrients delivered through new vessels. To achieve this, cancer cells can release signaling molecules such as VEGF, which instructs surrounding tissue to form new blood vessels toward the tumor. In laboratory studies, thymoquinone has been observed influencing pathways related to this process. Researchers have reported that it may reduce the expression of angiogenic signals like VEGF in certain experimental models. By affecting these signals, it appears to interfere with the ability of abnormal cells to promote new blood vessel formation. Again, this is not a clinical treatment effect. It is a biological observation under controlled conditions. But it is significant because it shows interaction with one of the key mechanisms tumors use for survival. The second major area of interest is apoptosis, or programmed cell death. In healthy biology, cells have a built-in system that tells them when to die if they become damaged or no longer function properly. This is essential for maintaining balance in the body. Cancer cells, however, often find ways to disable this system. They avoid death signals and continue dividing uncontrollably. This is one of the defining features of cancer biology. In experimental studies, thymoquinone has been shown to interact with pathways involved in apoptosis. Researchers have observed activation of proteins associated with cell death signaling in malignant cell lines under laboratory conditions. At the same time, many of these studies report limited or significantly lower effects on healthy cells under the same conditions. This selective behavior is one of the reasons it continues to be studied. It suggests that the compound may not act as a general toxin, but rather as a modulator of cellular signaling in different contexts. A third area of research involves inflammation and immune signaling. In cancer environments, inflammation often plays a supportive role in disease progression. Certain inflammatory pathways can create conditions that allow abnormal cells to survive and evade immune detection. One of the key molecular pathways studied in this context is NF-κB, a protein complex that regulates immune response and inflammation. In multiple laboratory studies, thymoquinone has been observed affecting NF-κB activity, which may influence how inflammatory signals behave in cellular environments. What makes this scientifically interesting is not that inflammation is simply blocked, but that its regulation appears to shift. The immune environment is not turned off, it is modulated. This distinction is important in modern immunology because both excessive and insufficient immune activity can be harmful. As these findings accumulated, researchers began testing thymoquinone in more complex models, including animal studies. In some of these experiments, tumor growth rates were measured under controlled conditions where the compound was administered alongside standard laboratory monitoring. Some studies reported reductions in tumor size or growth rate compared to control groups, while also documenting changes in molecular markers related to cell proliferation and apoptosis. However, these findings vary depending on dosage, model type, and experimental design, which is why scientists continue to call for more standardized clinical trials. At this stage, it is important to separate observation from conclusion. The presence of biological activity does not automatically translate into medical treatment effectiveness. That gap between laboratory research and clinical application is where most natural compounds remain under investigation. Still, what makes thymoquinone notable is not a single result, but the consistency of interaction across multiple pathways: oxidative stress, inflammation, angiogenesis, and programmed cell death. These are not isolated systems. They are interconnected networks that influence overall cellular behavior. And as research continued, attention also turned back to human studies, where scientists began examining how these effects might appear in real biological systems rather than isolated cells. That transition from laboratory models to human-focused research becomes the next critical layer in understanding why this compound continues to attract global scientific interest. We will continue from here in the next part. As interest in thymoquinone moved beyond basic laboratory experiments, researchers began focusing more on how its effects might appear in real human biological systems. This is where the gap between cell studies and clinical observation becomes especially important, because what happens in a petri dish does not always translate directly into the human body. In carefully designed human and clinical style studies, Nigella sativa extracts and thymoquinone have been examined for their influence on inflammation markers, immune response, and certain biological indicators associated with disease progression. These studies are still limited in scale, but they provide valuable insight into how the compound behaves in more complex systems. One of the most commonly measured areas is inflammation. In several small clinical trials, participants who received standardized black seed preparations showed changes in markers such as C-reactive protein, CRP, and interleukin levels. These markers are often used in medicine as indirect indicators of inflammation activity in the body. While results vary between studies, the overall trend in many published papers suggests a potential reduction in inflammatory markers in certain groups. However, these outcomes are not uniform, and researchers emphasize that dosage, duration, and formulation all significantly affect results. Another area studied is immune system balance. Some research has looked at T cell activity and general immune modulation after supplementation with Nigella sativa extracts. In these contexts, scientists have observed shifts in immune-related ratios and activity patterns, suggesting that the compound may influence immune signaling rather than simply stimulating or suppressing it in a direct way. This distinction is important. The immune system is not a simple on-off mechanism. It is a highly regulated network that requires balance. Too much immune activity can lead to chronic inflammation, while too little can reduce defense against disease. Most modern immunology research focuses on compounds that help restore balance rather than simply increase activity. In addition to inflammation and immunity, researchers have also explored metabolic and oxidative stress markers in human subjects. Some studies report changes in antioxidant capacity following regular intake of black seed preparations, including shifts in endogenous antioxidant enzyme levels. These enzymes are part of the body's internal defense system against cellular stress. However, despite these promising observations, scientific literature consistently highlights the need for larger, long-term, controlled clinical trials. Most existing studies involve small sample sizes or short durations, which limits the ability to draw definitive conclusions about long-term outcomes. This is a key point that is often misunderstood. Early-stage human research is designed to observe biological trends, not to establish treatment protocols. It provides direction for further investigation, not final medical recommendations. At the same time, researchers continue to study how thymoquinone behaves in combination with other biological compounds. One area of interest is its interaction with dietary antioxidants and plant-based nutrients. Some experimental work suggests that combinations of bioactive compounds may produce additive or synergistic effects on oxidative stress pathways, although this area is still under active investigation. Another important focus is bioavailability, which refers to how well a compound is absorbed and utilized by the body. Thymoquinone is known to be lipophilic, meaning it dissolves in fats. This property influences how it is absorbed in the digestive system and has led researchers to explore different formulations, including oils and encapsulated extracts, to improve stability and absorption. All of these factors, absorption, dosage, duration, and individual biology, play a major role in how any compound behaves in a real human system. This is one of the reasons why translating laboratory findings into clinical practice is such a long and careful process. What becomes clear from the human-focused research is that Nigella sativa and thymoquinone are not being studied as simple supplements or isolated solutions. They are being examined as complex bioactive agents that interact with multiple systems at once, including inflammation, regulation, oxidative balance, and immune signaling. And while no single study provides a complete answer, the accumulation of research across different institutions continues to build a broader picture of biological activity that is still being explored. This leads naturally into the next stage of understanding: how these findings are interpreted in practical contexts, how researchers approach dosage and formulation in experimental settings, and why safety considerations are just as important as potential benefits when discussing any bioactive compound. That is where we move next. As research into Nigella sativa and thymoquinone continued to expand, attention gradually shifted from isolated laboratory findings toward practical application frameworks. Scientists and clinicians began asking not only what this compound does in controlled conditions, but how it behaves when introduced into real-world biological variability, where diet, genetics, environment, and existing health conditions all interact at the same time. One of the first practical considerations in this research is dosage standardization. In laboratory environments, researchers work with precise concentrations of thymoquinone, measured in micromoles under tightly controlled conditions. But in human use, especially through natural sources like oil or seeds, the concentration of active compound can vary significantly depending on extraction method, storage, and processing quality. This variability is one of the main challenges in translating research into real-world use. A product labeled as black seed oil does not automatically guarantee a consistent amount of thymoquinone unless it is standardized and properly tested. That is why many scientific studies specify exact extraction methods or use purified compounds instead of raw plant material. Another important factor is stability. Thymoquinone is sensitive to heat, light, and oxygen exposure. Over time, improper storage can reduce its concentration and biological activity. This is why researchers often emphasize cold press extraction methods and protective packaging in experimental formulations. Even small changes in preparation can significantly influence the final chemical profile. Bioavailability is another key issue. Even if a compound is present in a product, it does not guarantee that the body will absorb it efficiently. Because thymoquinone is fat soluble, it tends to be better absorbed when consumed with dietary fats. This has led researchers to explore lipid-based delivery systems in experimental designs, aiming to improve how the compound enters systemic circulation. At the same time, human studies have shown that biological response is not uniform across individuals. Factors such as baseline health status, metabolic rate, gut microbiome composition, and existing inflammation levels can all influence how the body responds to bioactive compounds. This variability is one of the reasons why results in clinical studies are often mixed or context dependent. In several small-scale studies, researchers have observed changes in inflammatory and oxidative stress markers following consistent intake of standardized Nigella sativa preparations. However, these studies also highlight that outcomes are not immediate and typically require sustained use over weeks or months before measurable changes appear in laboratory markers. This brings us to an important scientific principle: time dependency. Biological systems do not reorganize overnight. Cellular repair, immune modulation, and metabolic adjustments occur gradually. Most studies examining natural compounds like thymoquinone measure outcomes over extended periods ranging from several weeks to multiple months. Alongside timing, researchers also emphasize the importance of combination effects. In biological systems, compounds rarely act alone. They interact with nutrients, hormones, enzymes, and other dietary components. Some experimental studies suggest that certain combinations of plant-based compounds may produce enhanced effects on oxidative stress pathways, although this remains an active area of investigation rather than a confirmed clinical strategy. Safety is another critical aspect that researchers consistently evaluate. While Nigella sativa has been used traditionally for centuries, modern science still requires structured safety profiling. Available data from human and animal studies generally indicate a favorable safety profile when used within typical dietary ranges. However, at higher concentrations or in combination with certain medications, interactions are possible and must be considered carefully. For example, because some studies suggest mild effects on blood pressure and blood clotting pathways, researchers recommend caution for individuals already using medications that affect these systems. Similarly, in clinical contexts involving serious medical treatments, any additional bioactive compound must be evaluated by healthcare professionals to avoid unintended interactions. These safety considerations are not limitations, they are part of responsible scientific practice. Every biologically active substance, whether synthetic or natural, must be evaluated within the context of individual health conditions and existing treatments. What emerges from this body of research is not a simple conclusion, but a layered understanding. Nigella sativa and thymoquinone are not being positioned as standalone solutions. Instead, they are being studied as part of a broader network of biological interactions involving inflammation, oxidative stress, immune regulation, and cellular repair mechanisms. And as scientists continue refining experimental models and expanding human studies, the focus is gradually shifting toward integration, how such compounds might complement existing medical approaches rather than replace them. This sets the stage for the final part of the discussion, where we bring together the research findings, practical considerations, limitations, and the broader scientific perspective into one complete view of what is currently known and what is still being investigated. We will continue into that final synthesis in the next part. When you bring everything together, the picture that emerges around Nigella sativa and its active compound, thymoquinone, is not one of a miracle cure or a replacement for modern medicine, but of a biologically active natural substance that continues to attract serious scientific interest across multiple research fields. Across decades of studies, researchers have consistently returned to the same core observation. This compound interacts with several key biological systems at once. These include oxidative stress regulation, inflammation signaling, immune modulation, and cellular repair pathways. Each of these systems is deeply connected to overall health, and none of them operate in isolation. One of the most discussed areas remains stem cell biology. Stem cells are essential for maintaining the body's ability to repair and regenerate tissues over time. As aging progresses, this regenerative capacity naturally declines, which is a normal part of biological aging. In laboratory settings, thymoquinone has been associated with changes in markers related to stem cell activity, suggesting it may influence how these repair systems behave under controlled conditions. However, translating those findings into real world clinical outcomes remains an ongoing area of research. Another major focus is cancer biology research. Here, thymoquinone has been studied for its interaction with processes like angiogenesis, apoptosis, and inflammatory signaling. In experimental models, it has shown the ability to influence pathways that cancer cells often rely on for growth and survival. These include blood vessel formation signals, programmed cell death mechanisms, and inflammation related transcription factors. Importantly, these findings come primarily from laboratory and animal studies, and while they are scientifically significant, they do not represent established clinical treatments. Human studies, though still limited in scale, have explored related effects on inflammation markers, immune balance indicators, and oxidative stress levels. Some of these studies report measurable biological changes over time, particularly with standardized preparations of Nigella sativa. However, results vary depending on study design, dosage, duration, and participant health conditions, which is why researchers continue to call for larger and more rigorous clinical trials. A consistent theme across all research is complexity. The human body is not a single pathway system, it is an interconnected network of biological signals, feedback loops, and adaptive responses. Because of this complexity, no single compound acts in isolation, and no single study can fully define its role. This is why the scientific community remains cautious. While early findings are promising in certain areas of cellular biology, they are not sufficient to establish medical recommendations. The transition from laboratory observation to approved clinical use requires years of additional testing, replication, and safety validation. At the same time, the historical and traditional use of Nigella sativa cannot be ignored. It has been part of various cultural and medicinal systems for centuries, long before modern scientific tools existed. Today, researchers are essentially revisiting this ancient knowledge through the lens of molecular biology and clinical science, trying to understand whether traditional observations have measurable biochemical explanations. What becomes clear is that this is not a story of replacement, but of integration. Modern research is not discarding traditional knowledge, nor is it accepting it uncritically. Instead, it is evaluating it through controlled experiments, biochemical analysis, and clinical observation to determine where it fits within modern medical understanding. For individuals, the most important takeaway is not to view any single natural compound as a solution to complex diseases like cancer or chronic illness. Instead, the focus should remain on comprehensive health foundations, medical guidance, evidence based treatment when needed, balanced nutrition, physical activity, and consistent preventive care. Natural compounds like thymoquinone may one day play a supporting role within that broader framework, but that role is still being defined through ongoing research. So when you look at Nigella sativa today, what you are really looking at is not a forgotten cure, but a scientifically active area of study that bridges traditional medicine and modern pharmacology. It is a reminder that nature still holds compounds we do not fully understand, and that science continues to uncover how deeply interconnected biology truly is. And that is where the story currently stands, not at an endpoint, but in the middle of discovery.